Getting louder and louder. No, it's all good. Okay, so here we are. Good afternoon. Been a great meeting so far. I had a great time listening to all those talks today. And some of them made me, well, hey, all of them made me think, darn it. But a couple in particular struck me as relevant to what I'm going to talk about, because I was sitting there thinking, why the heck am I talking about this stuff? And they have clarified things. So Katie's an educator. So am I. And I found that what I'm always doing is, my goal in these kinds of talks is for the audience to come away knowing something new. I want you to learn something from all of this. That's my goal anyway. And I was also listening to Matt, he's talking about debating all that stuff. I'm not a debater. I actually don't like debates very much because I think they're a bad way to communicate information. Again, being an educator rather than trying to dismiss bad ideas. But then I also thought about, how do you get people to listen to this stuff? So I'm going to tell you about molecular genomics today. Isn't that exciting? Yeah. It's always true. There's always somebody in the audience who's just enthusiastic about science. I saw hands go up. There are people who just love to hear about science. That's great. They're easy to get because they're already in the fold and they already listen. But then there's this other issue of how do we get other people to pay attention to this stuff? How do we make them listen? And I think debates are a good hook for that. That's what we're always looking for, is the hook. In my classroom, I get all these reluctant students coming in and saying, oh no, Myers is going to force me to learn about molecular genomics. How do you get them hooked? You have to know this stuff for the test. You have to know this in order to graduate. You're not going to get your degree unless you know this stuff. So that's the hook there. It's kind of a threat that you have to know this. But when I'm giving these talks, I'm always struggling. How can I engage people? How can I make them think about this? And one recipe, tried and true recipe that I always fall back on, is creationists are wonderful foils. They're people who can, I can show you the things they say, and then as the educational part, I show you how they're wrong, and that's always entertaining. Sometimes it's even funny. So that's kind of what I'm going to do today. Is I'm going to teach you a teeny tiny bit of molecular genomics, but I'm going to do it through this vehicle of exposing some ridiculous creationist ideas, which will be great fun. Okay, so that's my first challenge, is how do I get you to care about Synteny and these other things I'll be talking about. Then what I have to do is I picked something that I found hilarious when I read it, and I pulled it out and put it in a slide. And my next challenge is I have to show you why this is funny. So here it is. Go ahead, take a look at this. Oh, I heard nervous laugh. But, when I started reading this paper, I was just chuckling at the beginning, and then I was giggling, and then I was laughing out loud. It's such a bad paper. It's so ridiculous. And you're looking at this, and you're probably saying, what's wrong with it? It sounds very sciencey, doesn't it? It sounds really technical. It sounds like they've got numbers in there. It must be real science. They're talking about genomic. They're talking about genetic sequences and all this stuff. How come that's funny? So, unfortunately, that's a large part of what I'm going to do today is I'm going to explain to you why this is funny. And it never works explaining jokes, does it? I think what it really means is, I'm giving you a little taste, but what you really have to do is, I'm afraid all of you have to go back to school and get a biology degree. Okay, a few of you have already done this. You're ahead of the game. So that we can start handing you creationist papers and you can just laugh at them like it's an episode of the Jerry Seinfeld show. It'll work. Okay, so here's this paper. It's got all this technical stuff. And as I said, I read this. It was hilarious because every bit of it's just nonsense. But how do we know this? Well, one thing that I found really amusing right from the beginning is down there at the bottom, the authors, Bergman and Tompkins. And Matt will not like me for this. But I gotta say it, Bergman is an idiot. I'm sorry. You know, it's not fair usually. You know, there's a lot of intelligent people in the creationist movement. There really are. And there are people who are well-meaning and sincere. But I actually did a debate. My very last debate ever was with Jerry Bergman. And it was so painful, I never wanted to debate another creationist ever again. Because he's an idiot. I'm sorry. No, you know, I mean, it's not like he beat me up or anything. No, this was a total rout for Bergman in this debate. Afterwards, the creationists in the audience, they bust in a huge number of creationists from the churches there. They came up to me and apologized to Jerry Bergman and explained that he's not a good representative Christian. And I felt like saying, well, yes, he is. I can't be mean, I know. Anyway, so Bergman is kind of a nut. This is Jerry Bergman's bio. By the way, right now in Indianapolis, if you were there, you could be attending the Creation Evidence Expo. Aren't you lucky to be in Fargo? Where the featured speaker is Jerry Bergman, and here's his bio. Again, I'm bashing on the guy a little bit, but I just can't help myself. He's got 9 degrees, like it says, including 2 doctorates. Most of these degrees are completely random things. A couple of them are actually from unaccredited mail-order universities. He's very strange this way. You know, a couple of them are legitimate degrees, but he's been out there collecting degrees for 40 years. I don't know why. No serious scholar is going to, you know, maybe a couple if you've got disparate fields of interest, but otherwise this is really bizarre. Yeah, 960 publications in the science area. Most of that is in the Institute for Creationist Research's journals, if you want to call that science. And what I really enjoy about this is down here at the bottom, he lists all his entries in various Who's Who books. You remember these things? When you graduate from high school, you might have gotten a mail-in from them. Send us $40 and we'll include your name and the Who's Who. Yeah. He's a very strange man. You know, and the debate was not very pleasant. It was him accusing me of being a Nazi and things like that. Anyway, so there's one thing that made me chuckle is the authors. I know the authors, and I know that they're terrible. But the authors, the point they're trying to make, they're trying to use science to discredit something. And what are they trying to discredit? Well, it's an observation that was first made in the 1970s. Here it is. What are these? These are the chromosomes, diagrams of the chromosomes of 4 great apes. They're, you know, it's tiny, so you probably can't see very well. That's human, chimpanzee, gorilla, and orangutan. So 4 apes, and each of these lists the chromosomes. Okay, so there's chromosome 1, 2, 3, etc. And you can compare them and sort of see they're very similar. And this is noted, as I said, in the 70s. They noticed these deep similarities in the chromosomes, but they also noticed one significant disparity. And that is that all the apes, other than humans, have 24 pairs of chromosomes. And humans, also apes, have 23 pairs of chromosomes. What happened? Well, what's illustrated there, it's chromosome 2 actually, which is the oddball. And what you find is that there are 2 chromosomes in place of that 1 chromosome in apes. And when you line them up, like you can see here, this is Dean staining, which highlights various kinds of chromatin proteins that surround the DNA. You can see there's a banding pattern that's very similar. It looks very, very similar in structure. And you can take these 2 chromosomes from a chimpanzee, for instance, and you line them up, and they pair up very nicely with human chromosome 2. That's kind of cool, okay? We've got an example of similarity between apes, and we've got a difference, but we can explain the difference as a fusion event. That what happened is human ancestors had a fusion between 2 chromosomes from their ancestors to make 1 chromosome. We think it was a fusion in us, not a split in the others, because we're the oddball. Okay, all the other apes have 24 pairs. We've got 23. So you know, it's that exceptionalism thing. No, we're not exceptional. We're probably the product of the fusion. Okay, this is really interesting. It also led to some predictions. We can make predictions about what's going on inside the chromosome. So here's a simplified diagram of what happened. that there were 2 chromosomes in the other apes, in the ancestors of all humans, called 2A and 2B, and they fused. They just fused at the middle there. Now, what's interesting about that is that chromosomes have structure. There are details within chromosomes that you can identify. For instance, there is a centromere. That's a dark thing here. And that's a place where spindle fibers, these motor fibers attach to both chromosomes around the cell. And there are also these caps on all the chromosomes called telomeres. And telomeres have a very specific structure. Centromeres, unfortunately, do not have a very specific structure, so they're kind of harder to identify. But telomere is really obvious. I'll show you the structure in a moment. So anyway, what this suggests is that if there was a fusion event in our history, we ought to be able to not only see that they kind of line up with the other 8 chromosomes, but we ought to be able to look close up at human chromosome 2 and find telomere material in the middle of the chromosome, right? That makes sense. Well, that's an easy thing to predict. As I said, you'd also expect to find centromeres. There's little hints, but centromeres are so ambiguous that that's not good evidence. But finding a telomere in the middle of a chromosome instead of the end would be really interesting. Okay, so anyway, that's the prediction, is that we ought to be able to find that telomere there. So we've got questions now. Scientists love to reduce things to simple questions that we can then go try and answer. So there's the first really simple question. Where is the fusion telomere? Can we find it in modern chromosome 2 of humans? I'll get to these other 2 questions in a little bit, and that is, this is the one that the creationists forgot to think about. That is, what about the rest of the chromosome? Can we find similarities elsewhere? And the other is, well, if there was a fusion in our history, wouldn't it have messed things up? Wouldn't it have produced individuals that could never reproduce? And I'll answer all of these. So let's start with the first one. This is the big one. This is the one the creationists are very, very concerned about. That is, where's the fusion telomere? So a telomere, again, it's a cap at the end of each chromosome. At each end of the chromosome, there's this repetitive structure that's just sitting there. It has a very specific purpose. One purpose is that the enzymes that replicate DNA aren't very good at handling the ends of chromosomes. Any computer programmer types here? You've heard of fence-post errors, right? So a fence-post error is where you're copying an array and you're off by 1. And so you keep losing data from your array as you copy it. Biological enzymes have the same problem. The enzyme behind replication of DNA makes fence-post errors. It fails to copy the very ends of the chromosomes. So what has happened is that we have evolved a simple system to fix this, and that is you stick a whole bunch of junk on the end of the chromosome. So you just stick repetitive DNA on the end, so that if you lose bits of it, it doesn't affect anything. So what we've got at the end of every chromosome is that sequence right there. TTA, GGG, TTA, GGG, TTA, GGG, repeated thousands of times. Yes, the computer programmers here would say, what a kludge. Okay, every time I create an array, I'm just going to stick a bunch of garbage on the end of it, just in case I make an error in one of my routines. That's what's going on here. And I think that's also evidence for evolution. That's how evolution works. It's kind of sloppy and ad hoc, and it does what it needs to. This is also interesting because you see down here this little diagram, there's this complicated diagram. It actually has a particular sequence because it's going to fold over and overlap with itself and form double-stranded DNA with a chunk of itself down there. It actually folds over like a little paper clip. Okay, so every one of your chromosomes on the ends has this nice little thing where it takes the DNA, wraps it around like a little paper clip, or ties off the end of the chromosome to prevent errors from occurring. It's kind of clumsy, but it's brilliant. Okay, it works. It's a really nice thing to see. So telomeres are easy to recognize. You just see that sequence, TTA, GGG, and you see it many times, and you found a telomere. So a number of investigators a while back just said, okay, well, we've got these new fancy techniques. We've got all this data from the Human Genome Project. It's all on computers. Let's just go searching through chromosome 2 and see if we can find a telomere in the middle of chromosome 2, where it's predicted to be. And they did. they found a TTA-GGG sequence buried in the middle of chromosome 2. It has 158 copies instead of thousands. But again, that's not too surprising because, it's a result of a fusion. It was a collision between two chromosomes, and they lost bits and pieces in the process. So the thing is, though, that finding that 158 repeats is kind of dazzling. It's looking like, oh, that's clearly the signature of a telomere right there. Unless you're a creationist. If you're a creationist, oh no, we can't have this. Evidence that the chromosomes are the product of a fusion in ancient human history? That sounds like evolution. So they're busily trying to deny it. So for instance, here's the lovable Casey Luskin from the Discovery Institute. They wrote this big blurb about this where they were ranting and raving about it. He's saying the evidence for chromosomal fusion isn't nearly as clear-cut as people like to say. That, in normal telomeric DNA, there's thousands of copies of that TTA-GGG. But you see, what they found in the middle was only 158 repeats. Therefore, it must not be, right? And that's where we come into that piece that I quoted at you earlier, the amusing chapter or section from that paper. What this is the result of a paper by Bergman and Tompkins, who did the same thing those other investigators did. This data is all public. You own it. You can find it on the NIH website. You can download the entire sequence of human chromosome 2 anytime you want, all 237 million bases. What you're going to do with them, though, that's your problem. Anyway, so this is easy to get at. They downloaded all that 237 million bases, and then they had just had a little program, and they went searching for a TTA-GGG. Also, it's complement, CCC-TAA, because they got to have complementary pairs. And counted them. That 158, that's not so many. That's pathetic, because when they looked at the whole chromosome, they found 45,450 copies of that TTA-GGG sequence. Now you might be saying, okay, that sounds like numbers. All right, that's numbers all right, and 45,000 sounds like an awful lot less than 237 million. But it's still a lot. And 158 sounds pathetic. This is the entire intent of this paper, is to say, those 158 nucleotides, that's nothing. Just ignore those. They're inconsequential against the large number of these things that we find in the chromosome. And that's where these numbers, this is where I started chuckling at the numbers, is I sort of mentally did some calculations. I did pull out a calculator later and confirm that my mental calculations were right. And I thought, okay, 237 million bases. How many TTA-GGGs should we expect? What if it were a totally random sequence? What if the chromosome were totally random nucleotides? How many of those 6 base pair sequences should we find? And that's the thing you got to think about. The probability of any specific 6 base sequence in random DNA is 1 in 4,096. So you ought to see this every 4,096 base pairs, right? Just by chance. Remember, there's 237 million bases there? So 237 million divided by 4,096 happens to be 57,861. How many did they find? 45,000. Okay, that's in the right ballpark, I think. So basically what this entire paper is an analysis of the distribution of a sequence in the chromosome with no consciousness of the probabilities involved and coming up with random numbers that just sound big. So much bigger than that 158. So there should be 57,000, we saw 45,000. That tells you right there that these telomere sequences are depleted elsewhere, outside the telomeres where they should be. And so when you find 158, you should be dazzled and say, okay, that's unusual to find a lump of 158. Let me step back now. I want to mention one other thing. This is a tactic that made me laugh too. Notice that other number down there, 547,320. Where did that come from? He said there's a total of 547,320 internal bases on chromosome 2 that are composed of the intact telomere motifs. Duh, yeah. Yeah, when you think about it, they found 45,450 TTH-GTGs, they found 45,000 of the alternatives. Add those up, multiply by 6 because it's 6 base pairs long, you get 547,320. They can do basic addition and multiplication. Let's give the creationists a cookie. Okay? Isn't this marvelous? Anyway, so this entire paper is written casting doubt on the identity of this 158 repetition sequence. Utterly silly. It's irrelevant to the argument. What we really need to know is how often you find clusters, these islands of repeated telomere sequences. And no, they're rare, except on the end of telomeres. And finding one in the middle is really a good signal. It's really good evidence. Okay, but I'm not done. You know, the problem here is, as I was reading this paper, I was getting so focused on this evidence for a central telomere. And I was saying, okay, this is silly, we've got good evidence for this. But then I started thinking, why the heck do we care? Right? This is not the primary evidence for the fusion. Primary evidence for the fusion is synteny. Okay, synteny, I get to teach you a new word. Synteny means on the same band. It's a scientific thing. You see, if you've got a 1 3-syllable word, it's much better than 3 1-syllable words. I think it goes up by the cube with the number of syllables in the word. Anyway, so we've got this thing called synteny. What is synteny? Well, synteny, and I'm going to illustrate it with this lovely pair of chromosomes over here with a couple of genes on them. On chromosome 1, we have gene A and gene B. On chromosome 2, we have gene C and we have gene D. And synteny means on the same chromosome. So gene A is synthetic to gene B. Gene A is not synthetic to gene C. Gene C is synthetic to gene D. Man, we're talking kindergarten talking, and aren't we? This is really basic stuff. All that synteny means is that you've identified 2 genes or 2 sequences in the DNA as being present on the same chromosome. You're saying, what do you do? Okay, yeah, we kind of knew that there's bunches of genes on chromosomes. But what synteny leads, the concept of synteny leads to another interesting idea, and that's something called synteny mapping. We can make maps of synteny. What does that mean? I'm going to lead you through this step by step. So here we're back to our lovely pair of chromosomes here. This is in species X. The species X has 2 chromosomes with 4 genes. It's a really simple species, okay? And we have a related species over there, species Y. It's also got 2 chromosomes. It's got the same 4 genes, A, B, C, and D. But you notice they're in different arrangements, right? They're in different chromosomes. How can we illustrate this? What will make for a spectacular image that can be published in Nature magazine using as many colors as possible? I think that's part of the thinking. But anyway, it's a simple concept. So what we're going to do is we're going to take species X, and we're going to play paint by numbers. And we'll say, okay, let's paint the chromosomes in species X. So we'll say that all the nucleotides in chromosome 1 we're going to color red, and all the nucleotides in chromosome 2 we're going to color cyan. Okay, so a nice red and a nice blue for all the off to a pretty start. And we're going to use that as a paint by numbers key. Because then what we do is we pull out our fancy sequencing machines and we start sequencing the chromosomes in species Y. And we look for correspondences. Wherever we find the same sequence from this chromosome, for instance, this region here, we go looking for that over there in species Y, and wherever we find that sequence, we color it red, just like we did in species X. And wherever we find a sequence corresponding to something in chromosome 2 in species X, on species Y, we color it cyan. So we can do this is all done digitally, of course. We just got computers that can crank through this stuff, that can compare sequences from multiple chromosomes. They can say, okay, this chromosome we've colored red, this sequence is found here, 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 and other chromosomes, we'll color it red there too. And the end result is a lovely painting of the chromosomes in species Y, in which everything is color-coded by what chromosome it was on in species X. So you see how that works with this very simple example. And you look at that, now you look at species Y as color-coded, you might be thinking, how did it get rearranged? Do you have any hypotheses? You might imagine, well, maybe that region of the second chromosome that contains C broke off and was inserted into chromosome 1 in species Y. Seems reasonable. That there are genetic mechanisms that can do that, so we can actually map this out. So this is showing us in a nice, colorful way how these different portions of the chromosome match up to the other species chromosomes. Now, one of the simplifying factor is often what you'll see when you publish this is we've got a shortcut. We would just include species Y. But what we do is we put a little key up there and say that in species X, chromosome number 1 is what we're coloring red, chromosome number 2 is what we're coloring cyan. So we just get rid of this left half of the diagram and just show that right half. But what it does is it quickly illustrates how the chromosomes got juggled around in evolutionary history, right? You can see that. It just leaps out at you in bright, vivid colors. And just imagine if we had 23 chromosomes. We'd come up with 23 colors and we'd make a rainbow on the pages of Nature, or Science, or Cell, or wherever we aim to publish. So let me show you an example of this. I'm going to show you the chimpanzee Synteny map, where what we're going to show you is I'm going to show you the human chromosomes, and they've been color-coded by the color of, the arbitrarily defined color of the chromosomes in the chimpanzee. So here's that example right there. You notice anything about this? There are some regions where there's sparse information or there's a lot of repetitive DNA. That's easy. But notice that if you look at chromosome 13 of a human and you color-code it by the color of chromosome 13 in a chimpanzee, you find that it's all pretty much a big block of color there. And notice that there's not much of that dark green anywhere else. This is saying that chromosome 13 in a chimpanzee is pretty much identical in its sequence to chromosome 13 in a human. Are you beginning to get the idea of what the creationists overlooked when they're just looking at the telomere in the center of chromosome 2? They're ignoring all of this data, which says all the rest of the chromosomes are very similar as well. That's kind of a sin of omission if you're doing that. Now, further, this is where it gets really fun. This is chimpanzee human, okay? That's Synteny between humans and chimpanzees. We could ask this about other species as well. And we could do the same color coding, the same mapping. Now, what you have to imagine is that what's going on here is that over evolutionary history, we're seeing a slow shuffle of the arrangement of DNA and chromosomes, okay? Humans and chimpanzees, well, have been separated for 6 million years. And this is a slow process. So clearly, there hasn't been a lot of shuffling going on. Just a few quick shuffles, in particular of chromosome 2. If we look at something like a mouse, which has been separated from us for about 70 million years, what would you expect to see? This. Oh, yes, yes, get up and do an interpretive dance for us. No. Yeah, that you'd expect more juggling. That chimpanzees and humans have been separated for a little while. If you give them an extra 60 million years, then you'd expect even more variation. And that's exactly what we see. So over there on the right is the same diagram I just showed you for reference. That's chimpanzee Synteny. Here's mouse Synteny. A couple of things you ought to notice right away is, yeah, it's been juggled quite a bit. We've rearranged lots of the chromosomes. But another thing you might notice is that there's also these hugely conserved bits. Look at there, chromosome, this is, I can't read it from here, because it's all blurry. This chromosome, can you read it? No, I can't read it. This one right here, 25, this one? Yeah, I can't, anyway. Look there, this chromosome in chimpanzees and humans has nearly the same sequence, the same arrangement of genes. Similarly, this one, you see that big block of green, that says that region is pretty much the same between humans and mice. There is a little addition here, some of whatever chromosome blue was. Looks like chromosome 14 was patched on here. You get this idea that, yeah, there's a lot of similarity and there's also these differences. That's kind of cool. That fits with evolutionary theory, doesn't it? The farther apart they've been separated, the more the arrangement of genes ought to be scrambled. We can see this with a Synteny map. Okay, so that's a mouse, 70 million year separation. That's getting vicious. Let's look at an organism that's even more separated. Let's look at fugu, which is the Japanese pufferfish. And that's been separated from us for about 400 million years. What do you predict? Totally chopped up. Yeah, you expect lots of scrambling. You expect more scrambling than you would here. And that's exactly what you see, that it really gets finely broken up. You can still map these regions from humans to fish and vice versa. But you can see they're broken up into much more fine-grained bits. This is as if you've been shuffling for 400 million years. You can predict the cards would be pretty well mixed up by then. And that's what's going on here. Now, this is what the creationists ignored. Okay, I will agree with them that the telomere in the center of chromosome 2 is degenerate, it's short, it's not as long as it could be. It's been damaged by 6 million years of evolution. It was probably truncated when it was first formed. But why are you looking at that and ignoring the rest of the chromosome? Okay, maybe more ironic. This is a quote from David Klinghoffer, again at the Discovery Institute. And he's saying the same thing. Okay, that evidence from the human chromosome 2 fusion is ambivalent at best. He says, no, it's not. And he says, when you consider it in context of all the rest of the evidence, it becomes clear. The ambiguity is only really impressive when you consider all of the evidence. What are they smoking in Seattle? Okay, when you consider all of the evidence, when you look at the entire sequence of the chromosome, it becomes even more clear that chromosome 2 is the product of a fusion event. Yeah, so you know when I said Jerry Bergman was an idiot, there's a whole bunch of idiots over there, okay? You know, and what it is these are people, these are actually, you know, in a lot of ways, they're smart people. They've got a good tactic for making money over there, and they certainly hoodwink a lot of people. But the more you read the Discovery Institute stuff, the more you realize they know absolutely nothing about biology. They're completely clueless. They're making up as they go along. And so it's embarrassing when they come out and say these definitive statements like that it's ambiguous when you consider all of the evidence. No, it's less ambiguous. What I like to compare this to, here's what their evidence for chromosome fusion. And you've got a collision between two cars, okay? There's all kinds of debris all over the place. The Discovery Institute investigators come along and they say, oh, look at that little piece right there in the road. I don't recognize that. It's messed up. I can't, you know, I've seen something like this in a parts manual, but it was intact. It didn't have all these broken bits. Therefore, I'm pretty sure that there were not two cars colliding in this accident. And if you argue with me, I'm going to tell you you're ignoring most of the evidence. No, this is pretty clear. This is, you know, you've got lots of evidence. You've got the totality of these cars on both sides of the collision. There's some debris in the middle, exactly like you'd expect. This says there was a major fender bender here on this is the Creation Institute, in which you will announce that therefore this is an art installation in which these were created to look exactly like this in an instant. And that they've always been this way. Okay, so it's pretty silly. But now there is another question, a legitimate question. If we're saying that in human evolution there was a period when one human individual, the many times great-grandparent of all humans, had a chromosome fusion, so that they had a different number of chromosomes than all their fellow apes that they would mate with, doesn't that mean that they're going to be messed up and they wouldn't be able to reproduce and you would never see this fusion? That it couldn't possibly come around? This is something Bergman also says. He says these things like, they pose serious cytogenetic problems for the organism. Proper alignment requires the identical structure of each pair. Chromosomal fusion is one major cause of infertility, which is true. You do find instances of infertility caused by these kinds of changes. But again, I read this and I had to laugh. This is total nonsense. This is not true. They're exaggerating, misleading, and basically lying to you, unless they really are this stupid. But anyway, will it mess up mating? No, it won't. In principle, we will not try the experiment. Okay, there are rumors that some Russian labs have tried the experiment. You could breed a chimpanzee and a human together. What you would require is that, for instance, if it were the male providing the sperm, who would have a chromosome like this, you would have to have a pairing of these two chromosomes. In the chimpanzee, they'd have to align with this one chromosome during meiosis in order for this all to work. You know, that's true. That's what would have to happen. Is this a serious problem? No, it's not. It happens all the time in human beings as well. You know, it would be kind of cool to do the experiment with chimpanzees, except then you'd end up with these man-panzees, and you don't know what to do. Anyway, so it could work. And we know that in humans it happens all the time. This is my favorite example of this. I show this to all my genetics students because it's so mind-blowing. This is the result of an observation. It was published in the Journal of Human Genetics, in which a family came in for genetic counseling because there were some problems. For one thing, they had trouble having children, but they did have children. They had a daughter who had some slight problems. She was a little slow, she was a little mentally retarded, she wasn't doing well in school, that kind of thing. And they were wondering if there was a genetic problem with her. And they wanted to know what was wrong. And so they said, we'll just do a karyotype, which is where they take some blood cells and they flatten them out and they look at the arrangement of chromosomes, they count chromosomes. And they were shocked at what they discovered, not just in the daughter, but in the mother. The mother was weird. Okay, she had some very strange chromosomes. So what you see here is a result of the karyotype. This is the mother's chromosome. Notice here, this is chromosome 6. This is a fairly normal chromosome 6. Here's its complement. You notice anything funny? It's missing something. When they compared chromosome 9, there's one fairly normal one, and there's one, what the heck is wrong with this? It looks different than the other chromosome 9. Chromosome 11 is missing a bit up here. There are some other rearrangements down here. Chromosome 20 is all messed up. So, you know, my first thought would be, oh, Martians. The UFO nuts are right. There was a probe in it. No. This happens. You sometimes will have cytogenetic errors in replication. And I bet you some of you here in this room have little errors. Maybe not to this magnitude, but little errors in the chromosomes. Then they looked at the daughter. And she's got similar problems, but they're slightly different. Okay, so clearly what's happened is she's inherited some of the defective or damaged chromosomes from mom. She's inherited some novel defects of her own. There's been some rearrangements of these chromosomes that have been juggled around. It's very peculiar. They also looked at the father, but I'm not going to show you that because it's really boring because he was perfectly normal in his karyotype. So dad has normal arrangement of chromosomes. He's happily married to this woman who has a grossly abnormal arrangement of chromosomes. They have a little difficulty conceiving a child, but they had 2 children. It worked, right? So right there, we've got a refutation of Tompkins and Bergman, because these kinds of rearrangements aren't absolute obstacles to breeding. Over there at the far right, that's fluorescent in situ hybridization. It just color codes the chromosomes so that you can see more vividly the arrangements. Then they dismantled the chromosomes in a computer to figure out what their arrangement was. And here's a map of all the defects in the daughter. What you see here is the color coding illustrates these bits and pieces of other chromosomes that had been spliced in complicated ways to make the daughter's chromosomes. Weird, huh? And she's mostly normal. This girl was, like I said, she was a little bit slow. When they analyzed this more carefully, what they found is that she was actually the victim of a deficiency. She was missing a small piece of one chromosome, and that's why she was having the defects. Whereas the mother had the complete set of all the chromosomes, just juggled around in very strange ways. Okay, so that's kind of mind-blowing, isn't it? Then the question is, how could this happen? How could they get together? How could the mother and the father get together, and birds and bees and all that stuff, and arrange things? Because when they mate, when there's a fusion, you have to have a pairing of the chromosomes in order to get. Anyway, how could they do this? So here's what was going on in Mom. This is just a diagram we made of how her chromosomes must have lined up during meiosis in order to form a full complement, a full pair at every point. And so they had to do this kind of dance to juggle things around. It was an amazing game of Twister going on in her ovaries. But it worked. Took some juggling, but they eventually got all the chromosomes lined up and, for the most part, distributed them to the daughter as necessary. So that's pretty clear, right? Well, this is terrible. This is confusing. But it's pretty clear that, yeah, if you have cytogenetic errors, that's not an absolute barrier to reproduction. It does tend to have a greater frequency of progeny that are more messed up than the daughter, so you'll have a greater frequency of spontaneous abortions. So you could imagine that way back when, the ancestor of all humans had the fusion, and what would happen is he would mate with his partners who would not have been in the same arrangement. There would be a reduced fertility. He would have been lucky to reproduce. But once you got a small population of these people with the weird chromosome arrangements, something really interesting happens, and that now there is selection for reproductive isolation. Because now you will have more success having progeny if you marry, for instance, your sister or your first cousin or your second cousin. Creepy as that sounds. So what it meant is that very quickly, by cytogenetic means, this early population of humans separated themselves out and were less likely to mate with the other apes around them. And so this might have been an important reproductive isolation mechanism in early human ancestry. Okay, so let me just summarize everything. I'll give you all the answers here. So the first question I asked you is, Prince of Fusion time telomere is right where we expected it to be. There were no surprises there. This is what we expected to see. To argue otherwise, to say that, oh, it's just not big enough, that's really seeing the trees for the forest and missing the big picture, which is what's seen in the rest of the chromosome. When you look at the rest of the chromosome, they're a nearly identical match. So the fusion is pretty much undeniable. You can't argue with it. And does it mess up mating? Not really. There'll be some reduction in fertility, but not a huge amount. Okay. See, now do you understand why I started laughing when I read the paper? Okay. Read more Creationist papers, and you can laugh too, after you get your biology degree, like you all will. Okay, thank you.